Hello everyone, welcome back to my channel Bunny's Biology. Today we are exploring one of the most elegant editing system inside our cell, the RNA splicing mechanism. This is the molecular editing process that transforms a raw RNA transcript into a clean functional messages, prepare them for translation into protein. So this splicing process occurs in every eukaryotic nucleus and is carried out by a highly coordinated molecular machine, and that constantly editing and refining RNA transcript. So in this session we will examine this process in details. So before splicing begins, the cell produces a long primary transcript called pre-mRNA or hnRNA, that is heterogeneous nuclear RNA, through the process of transcription. So this RNA contains very different type of sequence. One is exon and another is intron. So what is exon? Exon are the coding or express segment of a gene that carry protein coding region. That means they contain the information required to build a protein. But intron are a non-coding intervening segment within the gene that interrupt the message and that are removed. So they are transcribed into initial pre-mRNA but do not remain in the final mature mRNA. So this is the mature mRNA, also called spliced mRNA. Here intron are non-coding sequence that are present between the exon. So this is intron, this is exon 3 and this is exon 4. This is intron 2 and intron 3. So intron are non-coding sequence present between exon. Because they do not code for protein, they are removed from the pre-mRNA by the process called splicing. But how does the cell know exactly where to cut? So in this mechanism, the cell identify intron using specific sequence signal that mark where each intron begins and ends. So every intron contains a built-in sequence signal that guide splicing mechanism, and this signal act like molecular signpost telling the cell where each cut should happen. So the key signals are: one is 5' splice site, another is 3' splice site, and other is branch point A. So 5' splice site is the boundary at the 5' end of the intron. This is the intron part and this is the exon part, and this part is called 5' end of the intron and also called 5' splice site, and this is the boundary between exon and intron, and this is the beginning of the intron. And next is the 3' splice site. This is the intron exon boundary at the 3' end of the intron, and this is marked by the 3' splice site, and this also intron and exon boundary. So another third sequence necessary for splicing is the branch point A. So the branch point A is a specific adenosine nucleotide located inside the intron. So it is a part of short conjugate sequence that called the branch point sequence, and that initiate the first chemical reaction. So it is found entirely within the intron, usually close to the 3' end, and is followed by a polypyrimidine track, also called PY track. This is the PY track. So the most highly conjugate sequence are the GU sequence in the 5' splice site and AG sequence in the 3' splice site and the A at the branch point. So the 5' and the 3' splice site were sometimes referred to as the donor and acceptor site. So these 5' splice site, 3' splice site and branch point A signal sequence that are present in every intron, and that specific signal sequence mark where each intron begins and ends. So after recognizing this sequence, the splicing machinery carries out two key trans-esterification reactions that cut out the intron and joins the exon. So the intron is removed through two successive trans-esterification reactions. So this trans-esterification reaction is the bond exchange reaction that do not require external energy. So in which phosphoester linkage within the pre-mRNA are broken and new ones are formed. So this is the 5' splice site and this is the 3' splice site. So the intron is removed through two successive trans-esterification reactions. One is lariate formation and second trans-esterification reaction is the exon ligation. So in the first trans-esterification reaction, it's triggered by 2' OH of the conjugate adenine at the branch site, and this group act as a nucleophile and that attack the phosphoryl group of the conjugate G at the 5' splice site. So let's see in the detailed structure. So this is the 5' splice site and this is the 3' splice site and this is the branch point that is present inside the intron. So in the first trans-esterification reaction, the 2' OH, this is the 2' OH of the conjugate A at the branch site that act as a nucleophile and that attack the phosphoryl group of the conjugate A at the 5' splice site. So the phosphodiester bond, this is the phosphodiester bond between the sugar and the phosphate of the 5' junction. This is the 5' junction and this is the 3' OH. So this is the phosphodiester bond between the exon and the intron, and this phosphodiester bond is cleaved. Then the free 5' end of the intron. So this is the 5'. So after cleaving this side, then this free 5' end of the intron is covalently linked. So this is the 5' phosphate, this 5' end of the intron is covalently linked with the branch point A and form a 2' and 5' phosphodiester bond. So this 2' OH covalently bond with the 5' free end of the intron. So cutting the RNA and forming a loop structure. This is the loop structure called a lariat. So a phosphodiester bond is broken and a new phosphodiester bond, this is the new phosphodiester bond is formed. So in the first reaction produce a lariat form and a free 3' OH. This is the 3' OH and this form a free 3' OH on exon 1. This is the exon 1. So in the second reaction, that is the exon ligation, the 5' exon is a leaving group in the first trans-esterification reaction. So in the second reaction, the 3' OH, this is the 3' OH of the 5' exon becomes a nucleophile and that attacked again the phosphoryl group at the 3' splice site. So in the 3' splice site, phosphodiester bond is cleaved. So here 3' OH covalently linked with the 3' exon site. So this is the exon 1 and this is the exon 2. So in the exon 1, 3' OH forming a phosphodiester bond with the exon 2. So this trans-esterification reaction has two consequences. First and most importantly, it joins with the 5' and 3' exon in which the two coding sequences are actually spliced together and create a mature mRNA. And second, this same reaction liberates the intron and which serve as a leaving group. So these two trans-esterification reactions occur during the splicing mechanism where the intron is removed from the pre-mRNA. So before we dive deeper, remember that cell actually use three distinct splicing mechanisms and each with its own strategy for removing intron. So one is nuclear pre-mRNA spliceosome that is mediated by the spliceosome found in all eukaryotes, and another two is group 2 cell splicing and group 1 cell splicing. So these both are cell splicing mechanism. That means both mechanisms remove intron without the spliceosome, but they use different nucleophiles and different RNA folds to carry out the same two trans-esterification reaction. So let's compare the three splicing mechanisms. So one is nuclear pre-mRNA that is present in most eukaryotic genes and that is very common. So these splicing mechanism work through two trans-esterification reaction and have branch site A, and this splicing mechanism mediated by the major and minor spliceosome, that is the catalytic machinery. Next is the group 2 intron that is rare and some eukaryotic genes from organelles and prokaryotes. The mechanism is same as pre-mRNA, and here is the catalytic machinery, that is the RNA enzyme encoded by intron called the ribozyme. Next is the group 1 intron that is rare and nuclear RNA in some eukaryotes, organelles gene and few prokaryotic genes. And this splicing involves two trans-esterification reaction, and here is the branch site is the G, that is the difference, and here the catalytic machinery is the same as group 2 like RNA enzyme encoded by intron, that is ribozyme. So in this structure we see the difference. In the pre-mRNA spliceosome mechanism, here is the A is the branch site, and in group 2 splicing mechanism, here the A is also branch point. But in the group 1 cell splicing, G is the branch point. And here is the others difference is the pre-mRNA and group 2 splicing mechanism form a lariat. But in the group 1 cell splicing, do not form a lariat structure. So now that you have seen the three different splicing mechanisms, so let's discuss the most common and most important splicing mechanism in eukaryotes, the nuclear pre-mRNA spliceosome or spliceosome mediated splicing. But first we need to understand what the spliceosome actually is. So spliceosome is a large ribonucleoprotein complex that is located in the nucleus of eukaryotic cell. So this complex comprises about 150 protein and 5 RNAs. So 5 RNAs are U1, U2, U4, U5 and U6 that are collectively called small nuclear RNAs, also called snRNAs. So each of these RNAs is between 100 and 300 nucleotide long in most eukaryotes and is complexed with several protein. So these RNA and protein complexes are called small nuclear ribonucleoprotein, also called snRNPs. So this complex is responsible for recognizing intron and exon boundaries and catalyzing the two trans esterification reaction, and that remove introns. But some non-snRNP are involved in splicing. One is U2AF called U2 auxiliary factor that recognize the polypyrimidine track in the 3' splice site, and branch point binding protein that also called BBP that bind to the branch site. Another is RNA annealing factor which help load snRNP onto the mRNA, and date box helicase protein that help in the spliceosome disassembly. So let's discuss the step of the spliceosome mediated splicing mechanism. So this is the pre-mRNA or hnRNA, and this is the 5' splice site, and this is the 3' splice site, and this is the branch point A. So initially the 5' splice site is recognized by the U1 snRNP using base pairing between the snRNA and the pre-mRNA. And next U2AF that is made up of two subunit. One is larger subunit that is called 65, another is smaller subunit called 35. So U2AF 65 and U2AF 35. So U2AF 35 that is the smaller subunit bind to the 3' splice site, and U2AF 65 that bind to the polypyrimidine tract PY tract. So the former subunit that interact with BBP and help the protein, that means BBP protein, bind to the branch site. And this arrangement of protein and RNA is called the Arley complex or E complex. So after that U2 snRNP bind to the branch site and aided by U2AF, and which facilitate the release of BBP from the RNA. So this arrangement is called the A complex. So base pairing between U2 and branch point create a mismatch. So this is the branch point nucleotide base, that is UACUAAC, and the U2 snRNP nucleotide are AUGAUG, and that create a mismatch, and this mismatches extrude the branch point A as a single nucleotide bulge. So now this bulge nucleotide, that is A, is unpaired and available to react with the 5' splice site. So next U4 and U6 snRNP along with U5 snRNP that joins the A complex, and together these three snRNP called the tri-snRNP particle, within which the U4 and U6 snRNP are stably associated through complementary base pairing between their RNAs. But whereas U5 snRNP is more loosely associated through protein-protein interaction. So with the entry of tri-snRNP, the A complex converted into B complex. So this rearrangement of the A complex to bring together all three splice site, and that form a loop-like structure. So in the next step, the U1 is leave the complex and U6 replaces it at the 5' splice site, and the base pairing between the U1 snRNA and the pre-mRNA is disrupted. So this loss of the pairing allow U6 to anneal to the same sequence. So this is the sequence where the U1 snRNP first bind, and after the U1 loss, the U6 anneal to the same sequence. So next is the U4 is released from the complex and allowing U6 to interact with the U2 through the RNA-RNA complex. This is the U6 snRNP and this is the U2 snRNP, and this arrangement called the C complex, and this U2 and U6 RNAs together form the active site. By the formation of active site, the 5' splice site of the pre-mRNA is brought into close alignment with the branch point adenosine and facilitate the first trans esterification reaction and form a lariate. And the second reaction between the 5' splice site and the 3' splice site is aided by the U5 snRNP, and which helps to bring the two exons together. So final step involve release the mRNA product and the snRNP with intron. So the snRNP are initially still bound to the lariate, but they get recycled after rapid degradation of that piece of RNA. But this spliceosome disassembly is driven by the DEAD box helicase protein called PRP22. So it is required for the second catalytic step of splicing and then remove the spliced mRNA from the spliceosome and allowing the complex to disassembly. So the splicing mechanism precisely remove intron and join exon to create a functional mRNA, and this accuracy is essential for the proper gene expression. So if this lecture helped you understand the mechanism, make sure to like, share and subscribe for more biology videos.